Neanderthal ancestry drives lipid catabolism in contemporary Europeans. So we have all heard of Neanderthals, but what exactly are they? Diverging from a common ancestor approximately 800 to 400,000 years ago, Homo neanderthalensis are the closest extinct relatives of humans, or Homo sapiens. Their genomes only differ by 0.3%. After first evolving in Central Asia and Europe, Neanderthals spread east into Asia. Between 70 and 60,000 years ago, contemporary humans migrated from Africa and spread across the continents. This leads one to believe that Neanderthals and modern humans could have coexisted before the Neanderthal extinction, which occurred thousands of years ago in Central Asian and European areas. Even though Neanderthals are extinct, some modern humans possess fragments of the Neanderthal genome that are specifically involved in the metabolic breakdown of lipids. Some scientists think that interbreeding occurred once humans left the African continent, which explains why Europeans have several sections of DNA in common with ancient Neanderthal samples. Another school of thought is that the similarity evolved independently through convergent evolution due to an advantage these variants in lipid catabolism offered. These shared regions are unevenly distributed across the genome, and some regions are particularly enriched with Neanderthal variants. For example, contemporary Europeans have as many as 3 times more Neanderthal variants in genes involved in lipid catabolism than Asians and Africans. Lipid catabolism involves the reactions and pathways that result in the breakdown of lipids and the release of energy. A possible explanation for this greater amount of Neanderthal-like sites in these catabolic genes is that it may have given a selective advantage to the individuals with the Neanderthal variants compared to anatomically modern humans that settled in the same geographical areas. The possible selection advantage has not been revealed thus far. A 2014 study assessed if Neanderthal variants resulted in adaptive lipid catabolism changes specific to Europeans. Scientists searched for regional similarities to the Neanderthal genome in the genomes of 11 contemporary human populations. 3 populations of African ancestry, 3 populations of East Asian ancestry, and 5 populations of European ancestry. For each of the populations, the human genome sequences were compared with Neanderthal and chimpanzee genome sequences. The Neanderthal genome consisted of a consensus of sequences from 4 individuals. Only single nucleotide sites displaying sequence differences between the chimpanzee and the 2 reference Neanderthal genomes were used in the analysis. A D-statistic test was used to express the mean difference between 2 human populations and their similarities in genomic sites with Neanderthals and chimpanzees. After analyzing the distribution of Neanderthal variants in the genomes of 11 contemporary human populations, the researchers found that genes involved in lipid synthesis contained a particularly high number of Neanderthal variants in contemporary humans of European origin, but not in Asians and Africans. Further DNA sequences revealed that the portion of modern humans that had migrated out of Africa into Europe share highly similar genomes with Neanderthals. This suggests that Neanderthals primarily interbred with humans in the European continent. These similar sequences show signs of recent positive selection, indicating that they give modern humans carrying the Neanderthal genotype a selective advantage to anatomically modern humans that settled in the same geographic areas. Currently, the assumed selective advantage is unknown. After analyzing the influence of Neanderthal variants on lipid processing in modern humans, the researchers further found recent evolutionary changes in lipid concentration and expression of metabolic enzymes in the human brains of European origin. The effect of lipid concentration changes on the brain is unknown, but the fact that Neanderthal variants might have changed human brain composition has interesting implications. This topic relates to our current module of biomolecules and metabolic pathways. This also relates to previous modules of evolution and ecology because this study includes examples of gene flow, niches, and species interactions. The topic presented 2 explanations of the similarity in lipid catabolism. The first hypothesis involves gene flow and demonstrates how hybrid zones can lead to stabilizing selection between 2 species. The second hypothesis suggests the similarity in lipid catabolism arose from convergent evolution due to directional selection in lipid catabolism.